Thank you very much, Wolfgang, for that lovely show. Thank you for those who nominated me for this prize and the committee for selecting our work, which of course has been contributed to by many members of my group and elsewhere. So also thanks to Brooker for making the prize possible. Unfortunately, there won't be any NMR in my talk today. But as Wolfgang said, I was trained here in Utrecht University, and it's great to be back home. I got lessons in NMR from Wolf, and I don't claim I remember much of it. But we are now doing NMR with Stefan Freund at the LMB using your magnets as well, and I kind of hope that Wolf's lessons still kind of make me not completely blind in those discussions that are ongoing. And that's because the main protein I'll be speaking about today, tau, is actually intrinsically disordered. So some of the talks you've heard before have shown what NMR can do for those. And so hopefully next time there'll be some NMR in the talk. So already, as referred to by Wolfgang, my group at the LMB is more or less split in two halves. So about half of the people work on prior image processing development in the Relan software, and then the other half of my group now works in very close collaboration with Michel Gurren's group on amyloid structures, and we've worked mainly on tau and alpha-synuclein, but we've recently also done some other ones. So just to briefly mention our image processing developments, although not the main topic of today's talk, they did make all that work possible. So Relan, we proudly develop open source, so we think it's very important in a field where there's ever more commercialization that we keep things completely open, so our scientists can build on each other's advances and the field can continue to progress very rapidly. So if you do follow me on Twitter, you may see sometimes I rant about how open software accelerates science. So last year, in collaboration with Thermo Fisher here in not far from here in Eindhoven, Abhay Kutecha there had access to a new prototype microscope with a cold field emission gun, a new Falcon 4 detector, and a stable Selectric X energy filter. And then Takanori Nakane, a postdoc in my lab, used this data collected by Abhay to calculate this apopharatin reconstruction to 1.2 angstrom, kind of reaching what is true atomic resolution, because each atom can now be almost all atoms can be represented by a single blob of density. I thought I wouldn't go into detail because you might have seen that. I thought just on the image processing side, I thought I'd give you a brief glimpse into what the future of our software will hold like. And I think, single particle cryo-EM really is a single molecule imaging technique, and thereby holds the promise to teach us about protein dynamics. And some of the previous speakers already kind of implied that how work on dynamics could feed into big deep learning frameworks like AlphaFold to not only predict static structures, but perhaps in the future also be able to predict how proteins react to perturbations, when other proteins bind, etc. So we're starting to think on the software infrastructure needed to do that, and we also have now turned to deep learning to do that. So Johannes Schwab, a new postdoc in the lab, is using variational autoencoders to train data sets of single particle cryo-EM images to train 3D deformation fields for each of these particles, and these kind of push around Gaussian blobs, which are our representation of the reconstructed density, hopefully describing for each of these images their conformation. And you can then do movies. And this is inner kinetic core complex from the Barford lab at the LMB, which was the consensus reconstruction of Relan was kind of limited in resolution. And if you just make movies of how everything wobbles with respect to each other, you'd see there's quite a lot of different states present in the sample. And then as a kind of validation that some of these dynamics are actually real, what Johannes then also did, not only you learn what are the deformations for all the particles, but you can then do real space weighted back projection type of reconstructions rather than the Fourier-based approaches Relan uses. And you can apply the inverse of the learned deformation to each of the back projected images, and you can then make a higher resolution reconstruction. So on the right here is a reconstruction made just in standard Relam with resolution more limited, and then by deforming all these back projections, you can make actual reconstructions of something that would probably be too small to do anything like multi-body refinement in Ryan to get much better density, kind of moving things forward. Now we're by far finished with that. The software is nowhere near being usable by other people. We're thinking hard about how to put as much prior information that we know about the distributions of density in real space into this, such that problems like overfitting and so on would be minimal. But I thought I'd give you a sneak preview there. So most of the work I'll be speaking about today was made possible by the PhD project of Sjors Scheres in my lab, who implemented helical reconstruction algorithms in Relam. And we used these then to work with Michel Goddard initially on this family of neurodegenerative diseases called tauopathies. So there's about 20 plus different tauopathies. These are all neurodegenerative diseases that are characterized by the abnormal inclusion of tau aggregates in different parts of the brain. And the most common tauopathy is Alzheimer's disease, abbreviated here with AD, and these are neurohistochemistry plaques on the pathologist, and you can see these tangle-like inclusions of tau in the cells. If you have different types of these diseases, for example, progressive supranuclear palsy or Pixley disease, then already at the cellular level, these diseases look very different, and also symptom-like. These diseases are very different from each other, but they're all characterized by the same protein going here. Now, tau in its normal happy life, if you like, is intrinsically disordered, as some of the other proteins discussed this morning, and then its function is supposed to be to bind to microtubules, where they are thought to stabilize the microtubules. And this fellow in Anogala's lab did a beautiful priam structure of some tau construct bound to microtubules, kind of giving us an idea how that might look like. But then somehow in disease, these intrinsically disordered monomers come off the microtubules and then aggregate into these very long filamentous assemblies, and here you can see a negative stain picture of one in the microscope. If you would put these perhaps in a X-ray beam, then you get this typical cross-beta diffraction pattern, where there's a very strong 4.7 angstrom signal in one direction, and then a lower resolution 10 to 12 angstrom signal in the perpendicular direction. And that is because these filaments are actually made of amyloids, where part of the structure forms beta sheets along the helical direction, and then because of the intrinsic twist of beta sheets, you get the twist of the filament, which makes the whole thing helical. So you get very long beta sheets all the way along here. That's not usually for the entire protein. It's something which you call the ordered core, where part of the protein forms a beta sheet, and these then pack against each other with a typical beta sheet packet distance of about 10 to 12 angstrom, which is responsible for that other part of the X-ray signal. Now, then, whatever residues stick out on the end and the C-termini of the ordered core, Tony Krauser, who looked at these type of filaments from tau people who died with tauopathies in the 80s, 90s already with negative stain, he called this a fuzzy curl. So this is just basically intrinsically disordered still parts of tau protein flopping out on either side of the continuous structured core. Now, in human brain, there's 6 different isoforms, and perhaps I wasn't trained too well enough, but before I wasn't too much interested in splicing and isoforms and so on, but it's actually quite interesting in terms of disease that in several tauopathies like Alzheimer's disease and chronic traumatic encephalopathy, you have a mixture of all 6 isoforms in these inclusions. But then it's of particular interest is these alternative slices of exon 10, because if that's not spliced out, you get the full-length repeats. The full-length sequence of tau contains 4 microtubule binding repeats, which Liz showed bind to the microtubules, and then you can splice out exon 10, you get 3 isoforms which only have 3 of these repeats. So these are called 3-repeat and 4-repeat tau isoforms. And for example, in Pick's disease, if you look at the inclusion, there's only 3-repeat tau isoforms in them. And a disease like progressive supranuclear palsy, there are 4 of them. There's actually a whole family of 4-repeat tauopathies, which are all characterized by 4-repeat tau isoforms aggregating. Now, this together with the observation that during the progression of disease, which can span decades, of course, you get what seems to be a spreading of tau pathology throughout the brain, then has led to this hypothesis in the neurodegeneration field that the spreading of tau at the underlying molecular mechanism might be similar to what happens in prion diseases, where you have filamentous protein and then you have intrinsically disordered monomers which are somehow convinced to join and make these filaments longer, and these can then break into multiple separate filaments, and you can get exponential spreading throughout the brain. How that would work was completely unknown, and what people had hypothesized was that perhaps these 3 repeats tau isoform composition of Pick's disease versus 4 repeat of PSP, for example, could be caused by different conformational strains, a concept familiar from the primary field. So how to study that was not entirely clear, but then with the resolution revolution, etc., and Sjors having implemented the helical reconstruction algorithms, we thought 2015-16 would be a good time to reassess looking at this in cryo. So we started from the brain of a 74-year-old female who died of advanced stages of Alzheimer's disease. And already Tony and Michelle showed in the 80s and 90s you can use sarcosyl solubilization of the tissue and then ultracentrifugation to purify these tau filaments from brain tissue and then look at them in negative stain. This is an example of a negative stain image from our brain. And you can see already described by Tony in the 90s 2 different types of tau filaments in Alzheimer's disease, what's called paired helical filament and straight filaments. And now, using our Titan cryo in Cambridge with the Falcon 2 detector, we were able to take images like this in cryo. You can still see the paired helical filaments here and the straight filaments. And we could now use Sjors software to solve to a resolution initially of about 3.5 angstroms the reconstructions of both the paired helical and the straight filaments. Now, paired helical filaments are called like that by Tony already in the 90s because they are actually made of 2 copies of tau protein molecule on each of the layers of the rungs of these beta sheets, if you like. So there's 1 copy here forming a C-shaped protofilament, what we call, and then there's a symmetry related, there's a helical 2-fold symmetry axis here. There's another copy of the C-fold, of the C-shaped fold in the paired helical filament. And it looked kind of wide, narrow, wide in the micrographs if you look here in the false colored one, because this structure is much wider than it is high. So this is an XY cross section along the helical axis. And the straight filament is made of the same 2 C-shaped protofilaments, but they're packed against each other in an asymmetrical manner. There's no symmetry relationship between them. And because this structure is more or less as high as it is wide, it kind of appears straight in the micrograph, and hence the term straight filaments by Tony. Now, 3.5 angstrom is then enough to build de novo atomic model in these, and Anthony Fitzpatrick, who did that as a postdoc in my lab, found out that the ordered core then spans residues valine 306 to phenylalanine 378 in the longest tau isoform, and could build a atomic model. Same for the straight filament. Again, these are the same structures. You can overlay them, and so there is basically one what we now call the Alzheimer's fold, and you have 2 types of Alzheimer filaments: the paired helical and the straight filament, where these 2 different protofilaments with the same fold come together in different ways. Now, the extent of the ordered core then explains why in Alzheimer's disease you have a mixture of both 3 and 4 repeat tau isoforms in the inclusions, because the ordered core starts at valine 306, so that's just after the second microtubule binary repeat, which is spliced out in the 3 repeat tau isoforms. So whether you incorporate tau isoforms with or without the second repeat, here shown schematically in orange, doesn't really matter, because that all sticks into the ordered core. So since that initial work which we published in 2017, we then went on. That was just one Alzheimer's case. So then Dan Falcon and Wenjun Zhang in my lab, they thought, what happens with other Alzheimer's cases, and this was a question we would get from the medics as well. You know, was this one lady, or is this something general? So in total, we looked then at about 15 different cases, and we did priam structures for 3 more of them, and we saw a negative stain for all 15, and cryo for the 4 cases. That you always, when you have Alzheimer's disease, you have paired helical filaments and straight filaments. Sometimes the ratios differ a bit, but it tends to be more PHF than straights. So we now think that the AD fault is general characteristic of Alzheimer's disease. Since then we've done many more cases, and this is always the case. So how about all the other diseases? I'll cut quite a long story short with this one slide. So since 2017, we've looked at almost all the different tauopathies. There's still a few relatively rare one left, so it's quite hard to get brains, but we're working on those too. And we've done structures of the tau filament extracted from brains of individuals with these diseases, always taking multiple cases of the disease, multiple individuals to make sure that indeed these faults are general, and that's what we've seen so far. And based now on all these structures, what you can see, these are now just schematic representations of that individual protofilament fold, would be the strands indicated with the arrows. And what you can see is that these structures are all remarkably different from each other. So I remind you, this is all just the tau sequence, but they can form into forming all these different type of tau filaments. And you can actually then do a structure-based classification of the diseases, where the mixed 3 and 4 repeat tau isoforms, Alzheimer's disease, but also chronic traumatic encephalopathy, the disease that you get if you have repeated head, that you may get if you get repeated head traumas. For example, if you play professional American football or boxing or so. Those are characterized by not having the first or the second repeat, and then the Pick's disease, which was the one which only had 3 repeat tau isoforms, does have the first repeat, because the second repeat is now spliced out as part of the other core. And then all these different 4 repeat tauopathies, among each other, many of them have different structures, but they're all characterized by the presence of the second microtubule binding repeat. Again, the extent of the ordered core explaining the composition of the tau isoforms in the inclusions. But of course, this raises an important question of what makes these structures different in all of the different diseases, because they're all made of the same tau protein. Now, before we go there, one old one that we found, we did in total 7 cases that had been diagnosed as progressive supranuclear palsy, or PSP. 6 of them had the same structure, but 1 of them was different. And we used Abhe's microscope again, got a 1.9 angstrom structure, and you can see the differences from the rest of the PSP structures, that, for example, obvious here at the C-tumoral part of the structure, the whole direction. But if you look in detail, there's all kind of small differences. Now, that kind of broke our whole observation of that different people with the same disease all have the same structures. So we then hypothesized perhaps this individual suffers from some rarer new disease that has not been described as a separate disease entity yet. So the neuropathologists went back into all the slices and back into the medical history, and they do think there is now evidence to suggest that this individual suffered from a new disease, which they then termed LNT, and so that kind of, we're suggesting what you can also do with prion is to identify new neurodegenerative diseases, which is kind of, as a structural biology, I find a cool thing. So now to that very important question, the same tau protein, this is not like a globular fold, which AlphaFold has now learned quite well to do. A protein sequence codes for a specific 3-dimensional structure in a globular fold, and that's what evolution has gotten to. That same thing is probably not true for amyloids. There are many different amyloid structures we've now observed for the same tau protein. But yet, if you take 10 different cases of AD, they all have the AD fold. You take 10 different cases of PSP, they will all have the PSP fold. So in disease, something very, very specific happens. I think it's important to understand the molecular mechanism behind disease to find out what those things are. So this is not only true for the tau protein. We've also shown similar things now for alpha-synuclein, amyloid beta, and others have used our software also to use this to show this for different diseases like amyloidosis by Mark and Strange, which is in Now, if we look into a bit of detail of the cross-sections of the reconstructed triangle densities, then besides the kind of beautiful main chain of the tau protein with its side chain densities, we also see all kind of different extra densities, which we cannot explain by the tau residues themselves. And they can happen either on the outside of the autocore or sometimes inside the autocore. For example, here in the CBD structure, corticobasal degeneration, there's something inside, and in CTE, there's also something inside. And I've blown these up a bit here. So CTE has a similar shape as the AD fold, kind of C-shaped, but it opens up a bit. And in the tip of the C, there is now a channel which is filled with a density that we don't, as of today, still do not know what it is. But it seems to be one of the major differences between the AD fold and the CTE fold, together with the flipping of in-out of two residues in the fold. And then in CDD, there is more positively charged channel with at least 3 lyses pointing towards a density which we hypothesize is kind of polyanionic. And now the question is, are these cofactors that are involved with the aggregation of tau? And if so, it would be really nice if we could find out what they are, because they might tell us something about molecular mechanism of disease. So, besides cofactors, it could be all kinds of other things. Of course, proteins get post-translational modified, and post-translational modification of tau, in particular hyperphosphorylation, is something that does happen in disease. So this might be another reason why the actual thing you're following up is chemically different, although it's always the same protein sequence. But then also things like, the actual isoform composition of the protein in a particular type of cells may play a role. Truncations, where proteases cut off pieces of the intrinsically disordered tau monomers, they could all play a role. Now, in order to study this, our post-mortem brain analysis has, of course, severe limitations, because this is only the end stage of disease, and it's a very non-perturbable system where I can't learn anything new from it. So, in order to be able to perturb system, I think we need to develop model systems of disease that can at least replicate the same structures that we've now observed in disease. And we're starting to do this in vitro, but also we're starting to do this also in cellular models, and even think about making new mouse models. Because Michel, for example, has a tau mouse model as well. We did the structure of that, again different from any of the structures we just observed in human disease. So how relevant a model for disease is that if the structure is different? So monomery full-length tau, if you make it in E. coli, is extremely soluble, and it won't come out of solution to make these helical assemblies. But if you add a bit of heparin, this was discovered by Michel and Tony in the 80s, then spontaneous, you get quite rapid formation of filaments in the test tube. Has been used by pharmaceutical companies to kind of have screens for high throughput screens of compounds that inhibit this type of aggregation, etc. But none of that was successful. And when Wenjun Zhang in my lab looked at the structures formed by the addition of heparin from full-length 4 and 3 repeat tau, she saw these kind of structures, again, very different from the ones we have observed in disease, in general quite a lot smaller, and we don't think that heparin-induced model system is a good description of what happens in disease. Now, it was only when Sophia Löwestam in my lab, who's currently a joint student with myself and Michel, started working on this problem, that we could make some progress. And it was by cutting off parts of the N and the C terminal sites of the fuzzy codes of the protein. And this was already known in the literature for many years. People have used truncated versions of tau that assemble very rapidly for many years. There's model systems based on concepts called K18 and K19. Unfortunately, they cut too much, so they cut into the order core of the AD fold, which makes it impossible for them to adopt the right structure. But Sophia followed up on a construct that was originally published by Louis Sertel, and that starts at 297 and ends at 391, and then she got structures like this following Louise's protocols, which again was not right, but she then tweaked conditions further. She played with how much you should shake, and the addition of a bit of magnesium or dextrin sulfate. She can now make, in the test tube, from recombinant tau constructs overexpressed in E. coli, bona fide Alzheimer's disease filaments which you can overlay with the ones from disease, and they're really the same ones. So again, we see some kind of extra densities that we also see in the AD ones, which probably indicate that they're not post-translational modifications, or at least not in the E. coli system. Perhaps in disease, I don't know, but they might be other things, like, for example, in these rats, we think they're just phosphate ions which stack in front of lysines, which are all on top of each other and positively charged. So there's a lot we can learn from this. We can now use this to do all kinds of experiments. And while doing this entire screen of looking for conditions to make disease-relevant structures, one thing that Sophia varied was the addition of salts, and she noticed if she added sodium chloride to the same buffer, she would not get AD fault structures, but she would get CDE fault structures, including a little bit of extra density in the tip of the C, where I've previously shown you this cofactor, potential cofactor density for CTE filaments was. She then thought, oh, what's so special? So she then changed the cation as well. She did lithium chloride and potassium chloride. And what she sees is you get different extents of opening up of this C-shaped fault, much like the difference between AD and CTE. So you can control this just by changing the cation of the salt in the buffer. And especially the potassium chloride structure, again using the microscope in Thermo Fisher, where we have a very beautiful collaboration still with APHE to do high throughput quorum, we were then able to do a reconstruction again to 1.9 angstroms resolution. And at that resolution, you can see individual blobs, 2 individual blobs per run in the tip of the C, which we kind of putatively modeled with potassium and chloride ions, because there's not much else in the buffer, and that the geometry of that is reasonable at least. So we now think that potassium chloride goes into that channel for the KCl structure, and then then if you look at the sodium chloride reconstruction that Sophia did, you don't get individual blobs, but you get kind of one elongated blob for each one of the beta sheet. And we can now putatively model that with sodium chloride ion pairs. So we think that sodium chloride, at least in our in vitro system, is the identity of that mysterious density at the tip of the C. Now, does that mean that in CTE, where there's density, we got quite pretty good resolution there too, 2.3 angstroms from the disease tissue sample, but there the density never separated into these elongated blobs. So we don't know whether it's also just something boring like sodium chloride in CTE, or whether there's something else going on. But I think it's an illustration that by tinkering with the system in vitro, we can actually start to gain understanding of how these kind of structures may form. While doing so, and this took her a bit over a year, Sophia managed to solve a lot of different structures, all of recombinant tau protein made in E. coli, just by changing construct length and conditions shaping salt, pH, etc. So in total, she managed to solve 76 structures in 1 year. About 30-something of them are actually new structures that were not yet observed before. And besides the AD and the CE structures I've shown you, none of them are relevant as far as we know yet for any of the diseases I've described to you previously. So that's just to show if you make proteins recombinant in vitro and you then do amyloid formation in vitro to do whatever structural studies, then that's fine. But it might be that the structure you make is not necessarily related to disease. Okay. So that brings me to my conclusion slide. I've shown you how amyloids are very unlike globular functional folds in that the same protein sequence can lead to a lot of different amyloid structures. So I think AlphaFold and approaches like this will have a pretty tough time at predicting any of this, which is great because you might all be out of a job, but I will still be able to do experimental structures. But why and how these different structures form in the different diseases, that is completely unclear. And I think that's where we really need to work hard on. And I think what we need to do is identify model systems little by little, start simple and build up complexity that at least generate the same structures as we observe in disease. That still wouldn't make it necessarily a good model system, but my hope would be that it would at least be better than a model system where that is not the case. So in this, we continue to have a lot of fun with what we now call high throughput cryo-EM. And high throughput, we kind of hesitate to put a number on, you know, because so many structures per month or week or day or whatever, I think is not so relevant because that changes all the time. I think the key here is that when I started out in Jose Maria's lab, you know, doing one cryo-EM structure was a PhD worth, and then now it has still become, I think, many projects doing the cryo-EM structures like the goal of the project. But this is now moving towards something where you run cryo-EM reconstructions more like, not quite as much as running a gel yet, but it becomes a tool which you can repeatedly apply to answer a wider question than just what is the structure. So in that terms, I think high throughput cryo-EM, we'll see a lot more of that, of course, in drug development, etc. There will be a lot of this, and we're very happy to collaborate with APE also on the hardware side and, you know, all the automated data collection tools they're doing there. So what hopefully will happen in the future is that knowledge that we gain both from these ex vivo structures and in vitro aggregation models or cellular models can then be translated into ever more complex, more similar to disease models, so that we can then have real tools in which to study, you know, the effects of potential therapeutics, which until now I think has been very, very difficult. So with that, I come to my acknowledgement slides. I'm very grateful, as already mentioned at the very beginning. You know, this work is all part of a very big team. So I'm grateful for all the members of my own group, past and present, who've contributed to the work I've presented you today and other work. All of the tau, alpha-synuclein amyloid work is in very close collaboration with Michel Goddard. So we jointly supervise all our students and postdocs. And if it wouldn't be for all these people on the right here who send us their brain samples or some samples purified from brain, then and of course the families of the individuals who've died of these diseases deciding to donate their brains, then none of this would have been possible. So I thank all of these people too, and I just put that in because to sneak it in because one of the previous speakers also made a pitch. So we're kind of carefully on the lookout for new group leaders in computational structural biology at the LMB. So thank you very much for your attention.